This presentation will cover the activation of B cells. My name is Amy and I will be covering an overview of B cells and their activation. B cells, along with T cells, are major cells of the lymphoid organs. These cells are known as lymphocytes. B cells are mostly found in the lymphoid organs but can also be found in the blood, although they mature in bone marrow. B cell functions include antibody production as plasma cells, becoming memory cells in order to fight further waves of infection, and also have a role in the activation of T cells. B cells are able to act on antigens through their B cell receptors. These B cell receptors are antibodies that are held on the surface of the cell. These receptors can internalise any foreign antigens that may be detected, and these can then be broken down into a variety of peptides. These peptides can then be represented onto the cell surface. These cells are known as thymus-independent cells.
Hi there, my name's Andrew and I'm going to be talking about class switching today.
Around me.
Class switching is a biological mechanism that changes a B cell's production of immunoglobulin from one type to another, such as from IgM to IgG. During this process, the constant region of the antibody heavy chain is changed, but the variable region of the heavy chain stays the same. Since the variable region does not change, class switching does not affect antigen specificity. Instead, the antibody retains affinity for the same antigen, but merely ensures a stronger response. This change is brought about via cytokines sent by the T cell. Once class switching has occurred, the stronger immunoglobulin will generally be able to clear up the pathogen quicker than it would have been able to before. Here's a quick picture about the antibody. As you can see, the big constant chain on the heavy chain is what changes and the variable stays the same. Alright, thank you.
Hi, my name's Sarah and I'm going to be talking to you about thymus-dependent B cell activation. This is where a B cell requires direct contact with a helper T cell to become activated. Thymus-dependent B cell activation occurs in the lymphoid organs such as the thymus or spleen. Specific antigens bind directly to B cell receptors where they are then degraded to peptides within the cell. The B cell acts as an antigen-presenting cell by displaying the peptide fragments on an MHC class II molecule on the B cell surface, which signal to T helper cells. When an appropriate T cell comes along, the MHC antigen complex binds to the T cell receptors, activating the B cell. The CD4 molecule on the T helper cell strengthens the binding. The CD40 ligand on the T cell engages with the CD40 on the B cell, releasing cytokine signals that induce proliferation of the B cell. The proliferated B cells differentiate into resting memory B cells and antibody-secreting plasma cells. The antibodies initially produce IgM, but as a result of T cell cytokines, they are able to undergo class switching.
Hi, my name is Bikram, and I've been asked to speak about thymus-independent B cell activation. So basically, what I need to talk to you about is how certain antigens can activate B cells directly. They can differentiate into plasma cells to produce IgM, which is a type of antibody. The antibody response is weaker than the TD activation. And lastly, really, is thymus-independent activation does not lead to memory B cells. Thank you.
Hi, I'm Paula and I'm going to be looking into the dental implications associated with B cell activation. So this is basically B cell mediated diseases. So as discussed by the others, B cells play an important role in regulating the immune response in both physiological and pathological conditions. However, dysregulation of B cell function can lead to severe consequences for the host. So the first one that I looked into was cancers. So this is mainly lymphomas. And you have two types of lymphoma. You have non-Hodgkin's and Hodgkin's lymphoma. Non-Hodgkin's is a lot more common and it's the most common hematological cancer in adults. It's by a clonal proliferation of lymphocytes which arise in the lymph nodes or associated tissues. The second type is a myeloma, which is caused by a malignant proliferation of plasma cells, so also linked to the B cells. Depending on the type of cancer, the treatment may be chemotherapy or radiotherapy. This carries with it a few dental implications, so The patient's more likely to have xerostomia, often taste alterations due to the chemotherapy and radiotherapy. They have a greater risk of caries and infection, and also oral mucositis. The second type that I looked into was autoimmune diseases which are associated with the B cells. So primary feature of autoimmune diseases is the loss of the B cell tolerance and the inappropriate production of antibodies. It includes multiple sclerosis, rheumatoid arthritis and lupus. B cell cytokines can play a role in other non-autoimmune inflammatory conditions such as type 2 diabetes and periodontal disease, which is often obviously really relevant for the dental practice, so you need to be aware that it's going to have implications for your treatment.